Okay, quick little jump in before the video gets rolling in its normal way. You've probably seen that it's a much longer video than normal. I've just finished recording it and I think it came to about 29 minutes, so it's probably definitely going to be over 30 minutes by the time I've done this prelude or intro. The reason it's a lot longer is I am talking about the origins of SARS coronavirus 2. Obviously, you've seen the title. That has become excessively controversial and conspiratorial and all those kind of things. So I really wanted to delve into it in a lot of detail and really lay out why I stress that this is a virus that evolved in nature. It's something that came from bats, possibly via an intermediate animal, and infected humans and started to spread in humans. It was not something that was made in a lab deliberately or accidentally. And I lay out all of the reasons I say that. So this came much longer, but hopefully it's good information and my arguments are laid out well enough to make sense. I've put the timestamps down in the description for the video, so if you want to skip to specific things, then that's hopefully easy enough to do. I do say this again at the end of the video, but to put it into my little prelude here, I'm expecting there to be quite a lot of comments and questions on this. There are some great questions that were asked in my previous video as well regarding the origins, and I've decided that I'm going to try and collate those all together and do one video tackling all of that separately. So if there's things I don't explain clearly because I go too heavily into the science, or if there's just any questions and things I don't tackle that you want me to, please leave comments down below and I will put that all together into my next video. Anyway, with that prelude said, here's the normal introduction to my videos. Okay, welcome to video number 25 of the Diaries of a Coronavirologist YouTube channel. Today is the 4th of June, we're now halfway through this crazy year, and we are around 6.6 million confirmed cases of COVID-19 around the world and about 388,000 confirmed deaths. So for tonight's topic, I'm going to be tackling something that's become a very contentious and controversial issue about the origins of this virus. So I have done a previous video talking about the origins of this virus before this was such a contentious issue, and I'll link that down in the description. But I wanted to come back and talk about it some more because obviously there's a lot of conspiracy theories and a lot of conjecture about how this virus came to infect the human population, and I figured I should talk about it. So at the outset, I want to very heavily stress the point that I am of the opinion that this virus evolved in nature. I think this virus evolved in bats and spilled over into the human population, possibly through an intermediate animal, or maybe directly from bats into humans, and it has then spread in humans ever since. I do not think that this virus was engineered in a lab or designed in a lab, and I do not think it was accidentally made in a lab and accidentally released. I'm very much of the opinion that it has come from nature, and I'm going to lay out those ideas for you in this video. So first up, I'll tackle just head on the idea of deliberately trying to make this virus, and I'm going to keep circling back to that as I go through some of the other ideas. Hopefully this doesn't get me into trouble with YouTube's terms of service or anything, because this is very much a hypothetical scientific discussion. So if you wanted to make a coronavirus that caused the damage that we've seen from SARS2, you take what you know about coronaviruses now, in that there's 4 that cause common colds and spread pretty well, but they don't cause severe disease. Then you look at the ones that cause severe disease, which are SARS1 and MERS, and you try and find some way to combine those properties into a virus that resembles SARS2. So SARS1 burnt out because it was too lethal, essentially. It caused very severe disease and only really spread at the peak of disease. So you could quarantine people and they would stop spreading because they're quarantined. As soon as symptoms appear, you put someone in isolation, and if they only spread when the symptoms are at their peak, they're not going to spread the virus. MERS, on the other hand, is even more lethal, but doesn't spread very well. So you want something that looks like a seasonal coronavirus and that it spreads and isn't super lethal and has some degree of lethality to cause the disruption we've seen now. Like I said, this is very hypothetical, taking the idea of if you were the big bad guy that people want to imagine there is, who had made this virus. You'd want something that looks like SARS2. You'd want something that spreads from asymptomatic people, has this silent spreading, causes a range of disease symptoms from mild to more severe, and can have lethality around that 1% mark, and therefore causes the disruption we've seen. The thing is, we don't know why that happens. We don't know why this virus spreads asymptomatically and SARS-1 only spread at the peak of symptoms. We don't know why the common cold coronaviruses only cause a common cold and don't cause more severe disease like SARS-1 and MERS and SARS-2. We have no idea why this virus spreads the way it does. So there is no way you could deliberately design this virus to do that. So just to approach that point head on, like I say, hopefully that doesn't get me in trouble with YouTube, because I've just come up with a hypothetical scenario where you may want to design this virus. Please, YouTube, watch the video in full to understand that it's a hypothetical scenario, and that I'm going to further debunk that as I keep going through this video. So now I want to just talk about some of the interesting features of SARS2 that have been discovered by looking at the genetic sequence. And they might be things that are important for how it spreads and how it's causing disease. We don't know for certain. We can only speculate that these things are playing a role in what's different between this and the other coronaviruses. But there are novel features that separate it from other coronaviruses that are interesting evolutionarily, and there are ways that they could have evolved in nature. There's also people who will argue that that might have been deliberately done in a lab, and I want to break down each of these parts and talk about why that's unlikely to have been the case. So first up, we have the receptor binding domain. And I've talked about receptors and the spike protein before, but just as a quick recap, the spike protein is the bit that sticks out on the surface of the virus. I always do this same little thing to remind you. And the receptor is what's found on the surface of cells. So the spike protein binds to the receptor, and that's what allows the virus to infect that cell. Lock and key is the analogy that always gets used. The cell has the lock, the receptor, the virus has the key, which is spike. So for SARS2, the receptor is a protein called ACE2, which is angiotensin converting enzyme 2. And spike must bind to ACE2. If it can't bind to ACE2, it can't infect the cell. Now, the most important part of spike for that binding is called the receptor binding domain, a very easy name, the RBD. So what's interesting about SARS2's spike receptor binding domain is it's very different to SARS1. So SARS1 was the coronavirus we knew about previously that causes severe human disease, that uses ACE2 as its receptor. And if you look at the SARS1 spike receptor binding domain and the SARS2 receptor binding domain, there's a big chunk of difference. So the receptor binding domain is 6 amino acids in SARS1 that are essential for interacting with that receptor. In SARS2, 5 of those 6 are different. So this is very interesting, because if you were to try and engineer this virus in a lab, you'd want to start with SARS1, since you know that can cause disease, and yet the receptor binding domain is very different. So like I say, it's 5 amino acids different out of 6. Now, if you were to try out all of the different combinations for that, that's 20 to the power of 6 different combinations to try and find this stretch of 6 amino acids in SARS2. So there's 20 amino acids, 6 different positions. That's 20 to the power of 6 combinations, which is 64 million different combinations that you'd want to try. So you wouldn't want to do that experimentally, because that's a lot of work. That's a lot of viruses to make, and that is a lot of research time. You would have to spend years and years with multiple people just doing that. It just wouldn't be done. You could, however, potentially computationally model it. However, the 6 amino acids in the binding domain of SARS2 are computationally predicted to not bind very well to ACE2. So computationally, this doesn't look like it should work, which is also what makes it very interesting. So you wouldn't come across this in a lab. You wouldn't design something that has this sequence of 6 amino acids that's found in SARS2, because you wouldn't predict that it would work, and you wouldn't evolve it, or you wouldn't try all of the different combinations. And if you were to try and evolve it in the lab, why would it change from, say, the SARS1 sequence? The SARS1 sequence works, there's no evolutionary pressure to change, especially to change 6 of the amino acids that are important. So this then brings me on to pangolins, which I'm sure people have seen hitting the news because there is a virus in pangolins that has been linked to SARS2. So a coronavirus was found in pangolins that has the same 6 amino acids of the receptor binding domain of spike as the SARS2 virus. The rest of that pangolin coronavirus is very different to SARS2, but the receptor binding domain is pretty much identical. So out in nature, there is this stretch of amino acids in the receptor binding domain that exists. Now, coronaviruses can have genetic recombination. So this is an idea of different viruses swapping packets of genetic information. The easiest way to explain this is with influenza, because influenza does this very readily, which is why we have had pandemics of influenza. Its genetic recombination is a huge driving factor. So with influenza, the genome is broken up into 8 different segments. So in the same way humans have different chromosomes, in a similar fashion, influenza has 8 genetic segments. And if two different viruses infect the same cell, they can swap those segments. So as long as you get a virus out that has all 8 segments at the end, it doesn't matter which virus gave which segment to that virus. So you can generate completely new viruses through recombination. Coronaviruses don't have a segmented genome. Their genome is continuous. But through complex mechanisms that I'm not going to go into in this video because it's going to be long enough as it is, you can have genetic recombination between different viruses. So since there's a virus in pangolins that has this receptor binding domain of 6 amino acids, we know it's evolved in nature, and we know recombination can happen between viruses. So if 2 viruses get into the same cell, they can recombine, and you can end up with a spike that has this receptor binding domain that we see in SARS2. But again, we wouldn't predict that this would make it work in humans, because we wouldn't predict it would bind very well to human ACE2. So the next feature to talk about is the polybasic cleavage site, or the furin cleavage site in spike. So just to do this very briefly, and I may do a longer video on it in more detail, but for coronaviruses to infect cells, they must bind a receptor, as I've talked about just now. The spike must bind to ACE2, and then either at the surface of the cell where that interaction happens, or within a compartment of the cell, the spike protein must be cleaved by enzymes, so protease enzymes. That cleavage allows for the spike protein to pull the viral membrane and the cellular membrane very close together to allow them to fuse, opening a hole through which the genetic material goes and gets into the cell, and that's when the cell's infected. So you need cleavage to allow this membrane fusion reaction to happen. Like I say, very brief, quick discussion, but this cleavage is very important. So SARS2 has this interesting cleavage site, this polybasic cleavage site, which is a furin cleavage site. So furin is a protease that can cleave that site. And so this is probably playing a role in how that virus infects cells. So there are coronaviruses related to the SARS coronaviruses. So this is called lineage B, to not go too deeply into it, but other lineage B coronaviruses that have polybasic cleavage sites, but they're not necessarily ones that are spreading super well in humans or spreading in humans or anything like that. So you wouldn't predict that this cleavage site would be playing a role in infection of humans. So again, if you were deliberately making this virus, you'd have no reason to think, oh, I should put in a polybasic cleavage site. Again, you'd look at the example that you already know of SARS1, which doesn't have this site. So why would you think to add one? Moreover, an interesting feature of this site is when you passage the virus in the lab, and I'm going to talk about passage a little bit more in a second, you see that site can actually mutate and be lost. It can mutate out and not be important. So when you're growing this virus in cells in the lab, you don't actually need that furin cleavage site anyway. So again, if you're trying to deliberately make this, why would you think to put that in? But as I say, this is already out in nature. We see it in other lineage B coronaviruses. So you can have this scenario once again of recombination, where there's viruses that have these features and they come together and they can swap genetic material and they can evolve. They don't even need to necessarily have that recombination. Just the fact that we have seen that this thing can evolve in nature points towards the fact it can evolve in nature, if you see that circular argument I'm making deliberately. So to come back to this idea of passage, let me just briefly explain what I'm talking about regarding passaging virus. So if you take cells in a lab and you infect them with virus and leave it for a few days, you can collect the virus that's come out of those cells, put that onto fresh cells, and repeat and repeat and repeat and repeat. That's passaging. You could do it in mice. You could infect some mice, you could collect samples from their lungs, infect new mice. You could do it in monkeys and whatever lab animal you choose. This is the idea of passage, that you grow virus, you collect the virus that comes out, and then you infect new cells and you repeat this process. And what this does is it drives the evolution of viruses to be good at infecting that system. So if you're working with cells in the lab, you are selecting, through natural selection, the viruses that are best equipped at infecting those cells in the lab. As I was just saying regarding the polybasic cleavage site, we find that if you passage SARS2 in cells in the lab, that cleavage site is lost. So it's possible that that's actually having a hindrance of the virus's ability to infect cells because you seemingly select for viruses that don't have it. So there has been a suggestion that maybe people were passaging bat viruses in human cells in the lab, and that's what's escaped, be it deliberately or accidentally. Now, we currently, and I'm going to again circle back to this, currently only know about the closest ancestor of rat G13 2013. So this is a bat virus that was discovered in 2013 that is 96% identical to SARS2. Now, 96% sounds close, and it is close, but it's not close enough to be a direct ancestor. It means they were related at some point. That 4% difference is about 1,100 amino acids. 1,100 bases of genetic material. And at the rate coronaviruses evolve, that suggests that there was a common ancestor between rat G13 2013 and SARS2 somewhere between 25 and 65 years ago. So they have common ancestry, but they haven't come from one. SARS2 hasn't directly evolved from rat G13 2013. In the same way, humans and chimps have a common ancestor, but humans haven't evolved from chimps. So the argument could be that people were passaging rat G13 2013 in the lab, and that produced SARS2 because they were using human cells and it evolved to infect humans. Now, the trouble with that argument is that when you're passaging in cells in a lab, you're selecting for viruses that infect cells in a lab. And cells in a lab are terrible representations of humans. And I say that as a cell biologist. So you're going to select viruses that are very good at infecting just those cells that you're working with. That's nowhere near the complexity of the human lungs. Obviously, this is a respiratory virus, the human lungs, the immune system that interacts with all of the different parts of what's going on in the body. Even our best cellular systems don't come close to the complexity of the human lungs. So it would be very, very difficult to evolve a virus to transmit well in the lab because you're selecting for viruses that grow well in the lab. And that's actually something that's done to make vaccines. You can make viruses weaker at infecting humans by passaging them in the lab, and then you have a live attenuated virus that can be used as a vaccine. So if anything, it would go completely the opposite way. Now, I will say there are more complex systems used in the lab other than cells. We use lab animals, we use mice, we use monkeys and things like that. But again, you run into the same hurdle that you're going to make a virus that's very good at infecting mice or that's very good at infecting humans. Sorry, not humans, monkeys. Let me try that one again. You're going to make a virus that's very good at infecting mice or very good at infecting monkeys if you are passaging in those animals. Now, while we're very closely related evolutionary to monkeys, we are still very different to monkeys, whichever species you choose. So it's just not something that can be done in a lab. Viruses evolve rapidly and they evolve to be good in the system they are in. So this idea that it was passage in the lab and either deliberately or accidentally, released, and that's where how we've got into this situation, once again, just doesn't make sense. What makes more sense is that this virus evolved in animals in nature. Then they started to come into contact with humans and had this fortunate ability that it could bind to human ACE2, and then it started to be able to evolve in humans in nature. I will do it. Maybe it was being passaged in humans in a lab. If you want to go there, be my guest, but I'm going to hope and assume that humans aren't test subjects in a lab anywhere in the world. Like I say, it's much more likely that humans came into contact with bats that were carrying these viruses, and that virus started to try and adapt to infect the human. Maybe the first one didn't get very far, but maybe that virus that's in bats then infects another human. It gets a slight mutation in the bat, affects another human, and then that virus can actually grow a little bit better in the human and can maybe go to another human, and you start to evolve a virus that can maybe spread. So the idea of passage in a lab just doesn't add up. It's much more likely, once again, that this evolved in nature and then came into humans and started to evolve in humans. And since I'm talking about this spillover into humans, another suggestion I've seen is that there could have been poor handling of the virus. So instead of it being a deliberate thing, it could have been accidental, and I've sort of touched on that a little bit just now. So again, that poor handling idea in a BSL-3 lab, or whatever is BSL-3, the Wuhan Institute of Virology, which is where people are saying this was done. That would require you to make this virus in the lab anyway. And as I'm saying, I don't know how you'd have made this virus in the lab. Now, potentially, to take this, to keep doing what I'm trying to do and look at the arguments that are being made and to tackle them, potentially, there's a virus that was being worked on in the lab that is 99% similar to, or 99.9% similar to SARS2, and that got accidentally released and it was never talked about. Now, that's just not how science is really done. So if you discover a new virus in bats, you publish it, you put the genome out there, you share this knowledge with people. That's the whole point of doing science, is you go, oh, look, I found something amazing here. So if there's something closer that we haven't been told about between that 96% rat G13 and the 100% SARS2 that we've not been told about in the 7 years since rat G13 and SARS2, that would require deliberate forethought of, oh, this virus looks like a virus that transmits really well in humans. How could you have known that a virus you were studying in that 7-year period would infect humans well? So why wouldn't you just publish it? If that makes sense. So you'd have had to know what you were targeting, and how could you know what you were targeting? So that idea that it was already in the lab because there's something that we haven't been told about, again, just doesn't add up for me. Now, again, there could be, and I have heard, the idea of mishandling at the point of collecting samples from bats. So here, the idea is that it did indeed evolve in bats, but then people sampling from those bats infected themselves with the viruses they were collecting. Okay, could happen. But the people making those sample collections dress up like they're going into a BSL-3. I talked about that in my previous video. You saw the extent of the safety kit we wear to work in a BSL-3, and people collecting samples from bats know that there's a potential that they could get infected and treat it the same way. So I consider it highly unlikely that a scientist would have been infected. What's much more likely is just that some person in China who comes into contact with a bat gets infected. So again, this is all stemming back to it's natural. It wasn't that someone mishandled it. It wasn't that someone deliberately made it. It is something that has naturally evolved. So like I say, right now we only know of the closest ancestor of rat G13, which is 96% similar. But we have only scratched the surface of the genetic diversity of viruses in general, but specifically here, coronaviruses, that are out there in nature. We need to be sampling bats more heavily, not less heavily, because the funding's been taken away. But, to stay on track, we need to sample more. There's probably viruses in bats that are 99.9% identical to SARS-2. We just haven't been able to discover them yet because we haven't sampled enough. I've laid out this scenario where we see that there's this receptor binding domain in pangolins and the polybasic cleavage site in other viruses, and so maybe they all came together and they evolved. A suggestion that I've heard made is that how would a pangolin virus come into contact with a bat virus? They'd have to infect one or the other animals. They'd have to infect the same cells at the same time to have that recombination occur, which is true. But like I say, we've only scratched the surface. I would be incredibly shocked if we don't find, well, if we can do the sampling, if we don't find within a few years that there are viruses circulating in bats that have the same receptor binding domain that's seen in SARS2 and seen in that pangolin virus. Viruses cover a lot of genetic space, and we don't even know the breadth of, we don't have any idea of the breadth of it. So I am so very strongly of the opinion that this evolved in nature. I don't think there's a way that it could have been deliberately made in a lab for the reasons I've laid out, and I don't think it could have been accidentally made in a lab either without us knowing more along the lines between, in that 7-year period, as I say, something would have been said, otherwise there'd have been the deliberate foresight to have the malice of making this, coming back to the deliberate idea, and there's no way you could have known that this would have been the virus that we have. So, hopefully I've laid out that I very much think this virus evolved in nature and spilled over into the human population, either directly from a bat or through another animal. And just to round that out, we have evidence for the fact that that can happen. SARS-1 was a virus that evolved in bats and then infected palm civets, had a bit more evolution in palm civets, and then became a virus that could infect humans. MERS, again, was a bat virus that infected camels, and from those camels infected humans. We have good evidence that coronaviruses can evolve to infect humans by being in other animal species. You don't need a lab. So while there are arguments being made that this virus was deliberately made or deliberately released or accidentally made or accidentally released or any combination of those things, for me it just doesn't add up. There's too many issues with that idea. I will say there are mistakes when it comes to working with dangerous viruses. So this idea of accidental release does hold some water. To give a few examples, there were cases of SARS-1 after the main spread stopped in China and Taiwan and Singapore that were connected to a release from a lab because someone messed up in a BSL-3. There were cases of smallpox that came from a lab after smallpox had been eradicated, so it could be traced back to the lab because someone messed up. Someone in Russia injected herself or himself with a needle stick of Ebola and died. The CDC sent vials of H5N1 bird flu to a lab that wasn't certified to work with them. Mistakes do get made. Accidents happen. It'd be wrong for me to pretend that these things aren't true. But like I say, I can't see any logical way or scientific way that this virus existed in a lab. The evidence is so much more in favor that it evolved in nature. All the bits are out there in different viruses. If we knew more about all the murky bits in between those viruses we do know about, we'd probably see something really similar to SARS-2. It's just an unfortunate consequence of viral evolution that this virus is here. It is not because of some malevolent approach to take down the world economy or whatever the story may be. And that's where I view this from as a coronavirologist, I guess. So I've done a lot of this in one take, which normally I try and do a few different takes, but it's quite a long one. I'm not going to do any of the questions. So people were great on my last videos, put questions down about this topic, and thank you for doing that. I have looked over them. What I'm intending to do is use those questions and comments, and what I'm expecting will be quite a few questions and comments on this video, and then going to do another video just on the questions and comments for my next one. So I'm going to compile it all together to approach all the different things. Like I said, I've done a lot of this one take today, and not gone back and edited and checked. So I may have explained things badly. And so if there's things that aren't clear, please leave a comment so that I can put that into my follow-up video or videos to try and further clarify things. I Might have gone heavier on the science tonight than sometimes I do. As I've said before, I try and make this accessible to everyone, regardless of scientific background. Um, but some of the explanations may have been not so good from me for the science behind the arguments I'm making. So again, I intend to follow up on this video. So please leave comments and questions, and I might open myself up to something. If there's other conspiracy theories that I didn't tackle, give me a laugh and put them down in the comments. But yes, please put the questions down there, because I will follow up and I will clarify things if I've made anything unclear in this video. I do really want to spread the information of why all the evidence is pointing towards this being a naturally evolved virus. And that's where I'll call time tonight. Like I said, probably a bit longer, this video. I have done a couple of cuts as I go through, but I haven't looked at the time together. But it's a big topic, and it was one I thought I should cover in a lot of detail, and it took me plenty of preparation time to actually film it. As always, if you're enjoying these videos, please subscribe, please recommend them to people, share them with people, drop a like on the YouTube, and leave comments and feedback and all that sort of stuff for the YouTube metrics. And as I always say, please stay safe, wash your hands, and keep calm and carry on. We will get through this pandemic. Thanks for watching.